Thank you so much for joining this seminar under EMSL Xchange Seminar Series. So glad to have Rhee here today as our invited speaker. Actually invited her once more than a year ago under the same series, but it was just a virtual seminar. Some of you probably remember that, but now we're so excited to have Rhee here, part of the in-person visit. So to introduce Sue, I should say that her CV is about 30 pages. So it's lots of contribution to science, lots of publications, grants. So this is just the first 7 pages, which I was going to summarize in 1 minute. Sue actually got a PhD from Stanford University in molecular genetic analysis of cell separation during Arabidopsis pollen development in 1998. So she joined Department of Plant Biology in Carnegie Institution of Science in 1994. And then this is where she still stayed, but she got several positions at the same institution. So she was also acting as a founding director for Arabidopsis Information Resource, or TER, which is one of the most well-known foundation in plant sciences, resource foundation, and while she was in the Department of Plant Biology in Carnegie. Then she also acted as a staff associate there, and then a staff scientist, acting director, and currently acting as a senior staff scientist in the Carnegie Institution for Science. So recently, she also she's acting as a founding director for a virtual institution called Water and Leaf Interface Institute, or WALI, which I think she's also going to talk about that today. So she has been contributing to science in different ways. If I just want to highlight some of those. Besides several grant proposals that she awarded to different organizations and scientific journal editor boards, publications, she's been mainly involved in establishing and enabling systems and omics biology. And in addition to that, she's been really active in revealing functions of unknown genes in plant systems and systems biology of plant metabolism and enabling metabolic engineering and transcriptional regulation and bioengineering. And very recently, she's been leading in the 4th single-cell plant biology, Plant Cell Atlas, which probably some of you are aware. So EMSL is also trying to collaborate with this very nice organization, which aiming to do a 4th plant single-cell biology. So with that, I would like to ask Sue to start the presentation. Thank you so much. It's all yours.
Thank you, Amir. Thank you for the invitation again. I think last time I gave the talk virtually, I talked about the Plant Cell Atlas project. Yeah. Today I'm going to talk about something different. And but before I start that, I wanted to say that there will be a Gordon conference next summer at the end of July in Ventura, California, on single-cell approaches in plant biology. Amir will be one of our speakers. There will be 21 other spectacular speakers, and the meeting will go on for about 4 or 5 days. And Gordon Conference, I don't know if you've attended it before, but they always leave the afternoons free. So you have sessions in the morning, in the evenings, and then there's a lot of opportunities to network. So especially for early career scientists, it's a wonderful place to really get to know people and present your work. So please consider attending that. So today I'm going to share with you a story that started with a grad student about 6 years ago. And as Amir said, one of the projects that we're very interested in our lab is to figure out systematically what the functions of these unknown genes are. These genes that are harbored in genomes of all organisms that have been sequenced. About anywhere from 25 to 75% of genomes encode genes that we don't know what they do. And they don't look like anything that look like they do. they don't look like anything where we know their functions of. So we sort of got involved with trying to figure that out in a systematic way. And this is the sort of, I guess, one path that we're taking with that.
So sorry, before, it seems like Mary asked you to turn on your video, the virtual attendant.
Okay. Let's see. I mean, it's not going to be.
It's gonna be
Great.
weird.
I mean, I can sit down. I don't know, Mary. It's not gonna. I could sit down. Does that work? Is that better?
Yes, that's better.
Okay, we'll do it this way. You're welcome. So this just shows you the pie charts of very heavily studied organisms. And the green parts of the pies represent genes that have been experimentally characterized. And the blue parts are those that are computationally predicted for function. And the gray parts are those that are completely unknown for function. It's probably hard to read the species names, but this is ordered based on how much is known. And the first one is Saccharomyces cerevisiae. Second one is human. Third is Arabidopsis thaliana. The fourth is fruit fly. This is mouse, zebrafish, and C. elegans. As you can see, each of these very well-studied organisms still harbors a lot of genes that are unknown for function. And the case is even worse for all of bioenergy models and crops. This information is actually available on a website that we just launched recently, and there's an associated bioarchive paper. Our intention is to keep this up to date, every 6 months. And this, again, I know you can't read this, sorry, the type is very small, but this is poplar on the left, and then brachypodium, wheat, I think, or wheatgrass, clammy, soybean, Ceteria italica, this is millet, sorghum, and so on. And so I just wanted to make a point that, you probably don't see any pieces of green here, right? They're very minimally studied, and still, even though a huge amount of investment is going into studying these plants, it's also possible that what's characterized are not captured in this sort of standardized functional annotations in the gene ontology system. Yeah, Chris.
So do you think, is this all data that is publicly available? Like for this?
This is all getting data from the Gene Ontology Consortium, Phytosome, and Uniprot.
So do you feel like maybe like glycine max, maybe there's some experimental data that the agro companies might not be making public?
Yeah, I'm sure. And there's a lot of data probably captured in the literature that are not yet extracted into these publicly available, easily, programmable annotations. But most of the genes are still uncharacterized. And so we wanted to see if we can push the boundary of functional characterization of protein encoding genes and try to see if we can identify putative transcriptional regulators using characteristics that are not sequence homology based, which is what the current standard is for function annotation. And so then what we did was take the set of unknown genes from Arabidopsis, group them into families based on sequence similarity, and then we use characteristics that we felt would be enriched in transcriptional regulators, things like being predicted to localize in the nucleus, having the ability to turn on the transcription of a reporter gene. And this is actually what's called autoactivators in these two hybrid assays. And then the high propensity of having disordered regions, which statistically is enriched in transcriptional regulators. And then we ended up with 43 families represented by 195 genes. And then we started characterizing a few of them. I'm not going to go into the other two families we're characterizing. Some of them have been published. This Chiquita has been published twice now, and we're working on a third paper. Flow is what I'm going to talk about today. This is a new gene that we found in Arabidopsis that seems to actually act as a water sensor and is involved in controlling germination of the seed. We also found a couple of families involved in forming stress granules in response to heat stress. I'm not going to talk about that today. Bottom line is, all of these families that we decided to pursue further in our own lab seem to be really interesting, novel regulators. I don't know whether they're really transcriptional regulators, but they're interesting regulators. And I think this set of unknown genes really represents a gold mine. And so I'm going to sort of tell you what we've been discovering from this one family. And it really took the bravery of a grad student, Yaniv Doron, in collaboration with a postdoc at Stanford, Stephen Boynam, now actually at the faculty at Baylor College of Medicine. And they actually established this enormous set of collaborators. And now the work is being led by a new postdoc, Sterling Field, and a research assistant, Jack Cox. But it was really a pleasure to observe this kind of new collaboration that emerged as a result of a lot of early career scientists talking to each other. So the first thing that we saw was that this gene, FLO1, is expressed most highly in the dry seed. And it was also very highly enriched in the intrinsically disordered regions. These regions that are really not amenable for three-dimensional sort of like structural determination by X-ray crystallography. There are so many of those crystal structures now that people can predict which regions of the proteins cannot be crystallized. So this is called disorder score, and you can see that this protein has two regions that are highly disordered. The one that's at the N-terminus that we're calling DS-rich region, and then the one in the middle that we're calling QPS region, okay, based on the amino acids that are enriched. Those were the two things that we knew about this protein going into this project. But we also knew that these proteins that are highly disordered and contain these prion-like domains can transition their physical states from being dissolved in the aqueous environment to being liquid droplets, to being kind of like gel-like state, as well as aggregated solid state. And some of these states can be reversible. And what we thought was kind of intriguing is that because this gene was mostly expressed in the dry seed, we were kind of intrigued about its possibility of going through different states, physical states, because the seed goes through these physical state transformations. When it's made fresh, it's mostly in an aqueous state. And then as the seed dries out during development and in storage, they're really dry. They're typically less than 10% water and reach a state that's called glassy state, okay? And this phase transition is considered to be important for the integrity of the seed and viability of the seed, but it's not really well understood how this happens. And also it's not well understood how the dry seed becomes aqueous again upon water, okay? What the biophysical process of germination is. So we were really interested to look at the biophysical properties of this protein and its role in germination. And so we asked a set of questions as follows. This is what I'm going to sort of talk about today. We first asked whether this protein phase separates, into these liquid droplets, and whether that phase separation happens in response to water. And we asked whether this protein is involved in seed germination and whether that seed germination is affected by drought or water stress. And we also asked whether this different states of FLO1 protein, whether it can modulate its function during germination. And then whether its expression is associated with the germination traits in nature across different ecotypes. And then we asked some additional questions that are kind of ongoing. the first 4 questions are the ones that we were able to answer, and that's what I'm going to share with you today. So before I go into showing you some results, I wanted to orient you on what you're going to be looking at. So this image on the cartoon on the left is the picture of a eudicot embryo inside a seed. So inside the seed, you have cotyledons here and here, and then you have what's called radicle, which is it's the embryonic root, okay? It's gonna develop into the primary root. And then I will be showing you a lot of these micrographs of cells in the embryo. And these kind of darker granules are seed storage proteins. They're autofluorescing. The sort of the darkness you're seeing is autofluorescence. And this blue dotted line indicates one cell, okay? And you can see that when it goes from, this is like in high salt or low water potential environment. And this is, oh, I guess you're not seeing what I'm pointing at, are you? The people on the Zoom can see it. The one on the right is when they're in aqueous condition, and you can see the autofluorescence goes down a little bit. So we first asked whether we can see these liquid droplets of FLO1 protein in the seed, and when we actually tagged FLO1 with GFP and examined the protein in the seed, we saw these granules. So this is one. Can't really.
Maybe you can use the pointer.
See if this is no. So this is like one seed, and one cell, and these dark points are FLO1 granules.
Touch these.
You got laser pointer?
Yeah.
Is that?
Oh, yeah.
Oh, now you can see. Thanks, that's great. Oh my God. When we actually dissected the seeds out in glycerin, in non-aqueous condition, we didn't see these granules. And then when we put them back into water, we saw the granules as soon as we put them into the water within a few seconds. And we can go back and forth how many times we could still see the presence of the granules in water and the absence of the granules without water. So this is reversible, like hydration or water dependent granule formation is what we saw with the protein. So that was kind of cool. But we didn't know whether this formation of the granules, how it depended on water potential. So we actually looked at the granule formation in the embryonic root or the radicle as a function of water potential using sodium chloride concentrations. And we saw this really interesting quantitative response of granule formation in response to water potential. This is like low water potential, high water potential here, and the low water potential here. What was interesting is that this granule formation at the cellular level was kind of all of the none, at the individual cell, but at the organ level, it was kind of like, this quantitative response. Okay, so this cell-to-cell heterogeneity sort of manifested itself into more of a quantitative response at the organ level, which was interesting. So so then we found that FLO1 basically forms these liquid droplets in response to water potential, or increased water potential. And so then we immediately asked, what is the physiological, first we asked them, what is the biophysical nature of these condensates? What are they? And so we expressed the proteins in vitro, and we can see them as these liquid droplets. And then we did these FREP assays to see how fast the proteins are sort of moving around inside the granules, whether they could recover basically by bleaching them. And we could see that they recover fairly well. So they sort of behave like liquid droplets. And then we asked, what is the physiological role of this protein? And in particular, what is the physiological role of this granule formation? And so we looked at germination in the knockout mutant, and we saw, so the black line here is the germination curve in response to increasing amounts of sodium chloride or decreasing amount of water potential. And so the black line represents wild type, and the red line represents the knockout mutant. So when you don't have this protein, the seeds seem to germinate more precociously under lower water potential. Okay? Wild type plants, they don't like to germinate when there's a lot of salt or mannitol or there's like low water potential. Okay, it's smart, right? But these mutants are less able to do that. Okay. And then what was really interesting was that the amount of the flow protein seems to correlate with this ability of the plant to germinate under low water potential. So each of these sort of circles represent lines that have different amounts of flow on protein. And you can see a nice negative correlation of germination fraction as a function of the amount of protein there is. This is actually looking at the protein level using GFP antibodies. And so there's this very interesting correlation between flow's ability to control germination under, low water potential. And so then we next asked whether this physical state of flow one, whether it can affect seed germination. And so now I'm going to go back to showing you the protein. I already told you that there are a couple of domains of these disorder regions. And so then we wanted to actually selectively remove each domain and see what happens to germination. First, let me show you what happens to the granule formation. This is showing you wild type. Now, this is actually transient expression of the protein in tobacco leaves. And so these kind of purplish areas represent a cytoplasm. Most of the cells in the tobacco leaves are vacuoles, which is represented by the gray here. And these dots, yellow dots represent flow granules, okay? So you can see in wild type, they form these nice granules. When you get rid of this first disorder region called the S-rich domain, we actually got this very large solid-like aggregates. And when we got rid of the QPS domain, we did not get any of this liquid droplet granules. So all the flow one protein was kind of dissolved in the cytoplasm. The DUF domain, domain of unknown function at the end, didn't seem to affect granule formation. So then the next question is, these mutants that we created, how do they affect seed germination? This is a complex figure, but I'm going to walk you through it. So the bottom here is basically how the protein's physical state is. It can go from being liquid droplets, like in wild type, or it could have no granules. In flow one, there's no protein actually, but in the QPS domain deleted mutants, there is protein, but it's not forming any granules. It's kind of dissolved. And then this plus wild type is the knockout mutant that has the wild type version back. And then this delta DUF is the domain of unknown function removed. Delta DS is the DS upstream, kind of like the disorder region that we call the DS region removed. Now, this y-axis is the seed germination. In wild type, under high salt, they don't germinate very well, only 20%, 15% or something. The flow one knockout mutants, they germinate much, much better, significantly better than wild type. When you put back the wild type copy into the null allele background, you get the same phenotype as wild type. It's complemented. When you get rid of this granule formation by removing the QPS domain, the phenotype is kind of like the null allele. Okay, so they germinate better. And then the DUF domain removal doesn't do anything. It looks like wild type. Also, its granule formation is not affected. What was kind of weird was this DS deletion mutant that made this like really gigantic solid-like fibrous structure. It germinated even better than the knockout. So it was actually acting as almost like a gain of function phenotype. We don't know what that means. I have some ideas, but this is what we observed. I think it's safe to say that the physical state of the protein seems to be involved in controlling seed germination under low water potential. And that weird DS deleted allele actually is no longer responsive to water in this granule formation. So it's like, in wild type, as I've mentioned before, under glycerin or under non-aqueous solution, you don't see these granules. When you put them in water, you see them. When you put them back into, like low water potential, they dissolve again. But these DS-less allele doesn't seem to respond to water potential. They're always there as the solids. So our current model is that flow 1 is acting as a resistor, and it's basically like preventing the seeds from germinating when the water potential is low. And it seems to be doing that through this physical state somehow. And where when we don't allow it to form these liquid droplets, they're not really able to resist very well. But then when we actually prevent them from being able to reversely form these phase separation bodies and have them stay at the solid state, they are actually like completely blind to water potential. Okay. And so this is sort of what we're observing. And what we would love to know is really like what is the actual molecular function of this, right? We're not quite there yet, but we have found some really interesting characteristics that I wanted to share with you. First of all, if this protein is involved in seed germination, which is a very important adaptive trait, you would think that its expression patterns might be somehow correlated with different sort of variation of seed germination in nature, right? So we took advantage of the ecotype genomes and transcriptomes available for Arabidopsis. And the reason why we started looking into this question is this. Flow1 protein gene actually naturally makes this DS-less allele as an isoform. So the gene is alternatively spliced and makes two forms of protein. The long form that I've told you about, and then the short form that's essentially missing that DS region. And as expected, that second isoform that we're calling 1.2 actually makes these very large solid-like structures. And what was also interesting is that the full version of the protein physically interacts and merges, dissolves each other with the shorter form. Okay, so the longer one is able to actually scoop up the smaller one, or vice versa. Okay, and so then we took the transcriptome data of the 1001 Arabidopsis ecotypes, and we looked for traits that were related to seed germination and asked whether flow1's expression is correlated with some of those traits. This is a trait that's called days of seed storage that's required to reach 50% germination. You can sort of consider it as a dormancy trait, let's say, and this sort of ability for the seed to not germinate from storage was positively correlated with the expression level of the long isoform of flow1. Okay, but it was the smaller protein, flow 1.2, that's missing the DS domain, is not correlated with that trait. Okay, however, flow 1.2, the smaller version of the protein, is positively correlated with precipitation in the summer and negatively correlated with maximum temperature in the summer. These are two traits that are related to promoting germination, actually. So it seems like flow 1.1, the longer version, is associated with dormancy or preventing germination in nature, and flow 1.2 is associated with promoting germination. So they may be, and I already told you, they interact physically. And so it might be that they're working together, to sort of modulate germination in nature based on the environmental conditions. Now, how widespread is this protein in the plant lineage? It's found in all green plants, including chlorophyta, green algae. Different numbers of homologs that's shown here on this graph. In the algae, there's only one copy. In flowering plants, there's multiple copies. And they all seem to make these interesting granules. But depending on the plant, the granule could look like the DS-less allele, like this here, or like here, or like here. Other plants, they make like wild type flow-1, like these small liquid droplets. And some plant, this is the Astrococcus tori, the protein goes to the nucleus, which is interesting. We don't know what the significance of the role of this protein homolog is in the plant lineage yet. But we think that studying these could play, studying these could actually like be useful for agronomically interesting traits, like, seed storage, how long seeds can survive storage in dry state, and also how seeds might respond to drought or desiccation. And so this controlling, this protein level might help us actually synchronize germination, perhaps. It's a practice that's actually used in agriculture. It's called seed priming, which is basically you kind of give some magic combination of solutes to dry seeds. You bring them up, you hydrate them, and then you dry them back out. And if you did that, the seeds will germinate much more synchronously and within much narrower temperature. Okay, and so farmers have been using this practice for a long time, and there are some companies that are selling these special concoctions, but no one knows what the underlying molecular mechanism of seed priming is. I think that flow-1's actually potentially got a role there.
And so this is exclusively expressed only in seeds?
Flow-1 is most abundantly expressed in the seeds, not very well expressed in other tissues. But flow-1 actually has homologs. I told you that like in plants, they occur in multiple copies, that they can be actually divided into two clades, flow-1 clade and flow-2 clade. Flow-2 clade is expressed everywhere. And it's expressed, and flow-2 clade is the one that's also found in the green algae. Okay, I'm going to actually show you this in more detail. I'm going to actually skip this part. But because you asked that question, I'll go right to that. So, yeah, so flow family exists in two clades, 1 and 2. And in Arabidopsis, the flow-2 clade has been duplicated. And so in Arabidopsis, there's flow-2 and flow-3. Interestingly, flow-2 and 3 also make these large solid-like aggregates, like the DS-less allele. Okay, as you can see here. And the flow-2 homologs are the ones that are more ancient and found in chlorophytes. And what's also interesting is that flow-2 and 3, the ones that kind of look like the DS-less allele of flow-1, they can really interact with each other very nicely. They merge, like you can see here down here. Flow-3 being tested with the DS allele of flow-1. And then this is like flow-1 sort of interactions with flow-1, which mixes fine by itself. With flow-2 and 3, they kind of interact, but they don't mix, right? You can observe them as sort of separate entities in the granules. And the DS-less version of flow-1 behaves very similarly to flow-2 and 3. And I'm not showing it here, but flow-2 and 3 mix very well with each other. Like this is how flow-2 and 3 would look like, okay? So it's maybe possible that in the seed where all 3 proteins exist, that flow-2 and 3 might be acting to sort of absorb flow-1. And so then when you actually have this, and they're kind of like playing possibly opposite roles. And when you have this DS version of flow-1, the flow-1 is no longer able to kind of like play that opposite role because it behaves like flow-2 and 3. And so maybe that's what the gain of function-like phenotype of the DS allele might be in germination. And so, yeah, we don't yet know what the phenotype of knocking out all these 3 genes would be, but my guess is that it's going to behave similarly to the DS allele. That's what we're going to be testing soon. And so finally, what the molecular function is, we are actually looking at viscosity changes in the cells of the seeds using sort of biosensors called GEMS. These are sort of fluorescent proteins that's hooked up to viral particles that are of exact sort of same size, and they can go into different numbers of molecules or subunits into the particle. They're basically like fluorescent nanoparticles that can be genetically encoded, and they're called GEMs. And so we've actually put these biosensors into Arabidopsis wild type and mutant lines, and we're looking at the viscosity changes in the seed as they germinate and as they dehydrate. And it looks like there's actually quite a bit of changes going on in the diffusion rates or viscosity of the cytoplasm. So we're actually moving into looking at biophysical parameters of the cells now. And so now I'm going to actually sort of spend a few minutes describing this new institute that we started that Amir talked about, WALI. We basically, this institute is essentially the set of people that collaborated with Yaniv and Stephen in looking at FLO1 protein. And we were actually really happy collaborating and wanted to continue our work. And so we wrote a grant to NSF to form a biology integration institute. We're calling it Water and Life Interface Institute, or WALI. And we brought in a few additional scientists here, but basically we're a group of 11 labs that's distributed across the US. And we're really interested in studying what's called anhydrobiosis, or life without water. And so, most biology happens in the context of water, right? All of our tools, everything you guys do here is based on aqueous solutions, I think. Well, I might be wrong. But there are some organisms and cells that can withstand not having water and survive desiccation. This includes seeds, of course, but also pollen. Some microanimals like tardigrades, phases of nematodes can actually desiccate tolerance, can tolerate desiccation. And fungus like Aspergillus actually thrives under desiccation. They will actually grow. This is why Aspergillus is such a bad storage food spoiler, because they will actually target dry seeds and grow, in a desiccated state. And of course, some resurrection plants. No one really knows what the molecular mechanisms of desiccation tolerance is, and how these cells and organisms can really not only survive desiccation, but how do they revive from being desiccated? What are the molecular and physiological processes that go on? That's what we're trying to study in this institute. So we already have a website. It's called walii.science. And we have a Twitter handle. It's called walii_nhydro. And it just started a couple of months ago. And so, we're just about to get started with hiring folks and so on. But we're going to study this anhydrobiosis in 4 different themes. One is to study physical and cellular properties of anhydrobiosis, looking at things like biophysical, biochemical, and ultrastructural characteristics. We're also interested to study the physiological recovery of desiccation in seeds and pollen and so on. And we'll look at their physiology, omics, properties, live imaging, and so on. And then another theme is to engineer resilience to anhydrobiosis by looking at protein structures and amino acid compositions, and using AI and synthetic biology. We're going to ask what properties of proteins will allow cells to withstand desiccation and would allow molecules to withstand desiccation. Finally, we're looking at the evolutionary dynamics of desiccation tolerance by comparing sensitive to tolerant pairs of organisms, or looking at ecotypes across species using quantitative genetics, ecophysiology, and genomics approaches. So I'm close to ending my talk. I just want to end by saying that even though anhydrobiosis is not very well studied, it really was responsible for starting civilization as we know it, because the ability for people to store seeds was what led to the birth of towns and cities. And of course, today it's very important in terms of looking at climate change and how we can sort of engineer or breed resilience to water limitation in seeds. It's very important for ensuring viability of grains in a dry state. But going beyond that, this idea of anhydrobiosis concept and mechanisms could potentially help us think about alternatives to cryopreservation. It's called zero preservation, preserving things in dry state, or the possibility of eliminating coal supply chain for proteins, antibodies, vaccines, and possibly even green chemistry that we could do without aqueous solutions. So I want to end by thanking all the people that have been involved in this project, and now all the people that are involved in the WALI Institute. I want to especially give a shout out to this group called Intrinsically Disordered Protein Scientific Interest Group, or IDP-SIG, which is a kind of a grassroots organization that postdocs and grad students at Carnegie and Stanford formed a few years ago. And it's through this sort of group that had these seminars on a monthly basis that we were able to meet most of our collaborators. And that group is still going. They're doing virtual webinars and so on. And I just want to highlight the importance of these sort of grassroots communities. Well, thank you so much. That's what I have for you today. I'd be happy to answer any questions you might have.
Any questions on that?
Can I have a quick question? I'm more like on the structural biology side of things, and I was just curious about this body formation. Do you know specifically if it's forming these bodies by itself, or it needs some other additional protein factors to come and play? Do you have any information regarding that? And
yeah,
like in general,
yeah.
Like,
oh, sorry. Continue.
so does it form the bodies by itself, or does it need some additional factors to form those bodies? Do you have any information about that, or not yet?
Yeah, I do. It's like I don't want to escape because it escapes. So
There's you mentioned co-expression, like
yeah.
that some of them you need to have like
Yeah,
they come together, but beyond this class of proteins,
Yeah.
do you have any other helpers there?
you don't need helpers. So this actually is in vitro.
Grains in vitro expressed.
Yeah, so we expressed it in E. coli, and I isolated it. We isolated it. It's actually done by a grad student, Eduardo Flores, in Shahar Sukenik's lab at UC Merced. They expressed the protein, purified it, and then did this assay.
Did you do any structural work on them?
So we would love to do cryo-EM stuff. Now I will actually escape, and then I'll have to re-share my slides. But we've done some correlation microscopy and TEM. I think I actually have some TEM data here. Let me see if I need to share again.
Because we do have appropriate capabilities to do like a lot of things that you're of interest to you. We have like single particle cryo-EM, and in terms of like different proteins, we can do co-expression of proteins because we do it in a cell format. It's easier.
Yeah, no, I think it would be really, and this is a very interesting protein to look at more carefully using fancy microscopy techniques. This is TEM. This is what the liquid droplet looks like. This is in human cell lines, actually. And then this is, remember I told you the solid-like state of DS-less mutant? This is what it looks like at ultra-structural level. It kind of looks fibrous. You can see this potential rods going in. We would love to see this, maybe do cryo-EM,
Because we have
ET.
cryo tomography.
Yeah, cryotomography would be great. We have tried to do some cryotomography with Marissa Otegi at Madison, Wisconsin. But yeah, I think we haven't done it in the embryo. Well, no, we've done it in the embryos, but not in the not in this mutant. I think we've done wild type. And we didn't quite see them well, actually. But this mutant is the one that would be interesting to do. Yeah.
Yeah, very quick comment. Yeah, that's very. I'm also like a very interested in structure, and I know like a native mass spec field has used kind of the approach to look at like amyloid fibers like those. So there's some information we can probably get with some of the domain,
Yeah.
and you can look at like early aggregates and
Yeah.
get some biophysical properties and see how they form.
I think that would be really great. And now we've figured out a way to control water amount during germination. So by putting the seeds in under 95% relative humidity, we can basically expand the first hour of germination, by which time like everything's happened. So like, but then we can expand that over a course of 3 days. So having them in this special high humidity chamber, the dry seeds, over 3 days is the same as putting them into aqueous solution for an hour. So we are now able to actually get to that early stage of hydration that we were not able to see before. So yeah, I think there's a lot of really cool things we could do because we can capture those embryos, right? And then send you the seeds like different states. Yeah.
So I think I remember hearing that GFP tends to clump, or there's clumpable GFPs and non-clumpable GFPs. I mean, is there any influence of the GFP tag on, do you think, on some of the behavior of the protein, or how can you separate that?
Oh, that's a good question. That is actually an interesting question because all of the live imaging I've shown you is through GFP. Even this is GFP tagged, okay? Because this was also done through fluorescence correlation microscopy. And the ones that I told you about that we're working with Marissa, they were not GFP tagged, and we couldn't see these liquid droplets very well in her setup. So I actually don't know. I don't know right now whether this non-GFP version, how they would behave. That's a really interesting question. We are observing changes in viscosity of the cytoplasm. In a cell that's like not GFP, doesn't have a GFP sort of, actually doesn't have flow GFP, but it has, of course, the GFP, that's the biomarker, the nanosense, the nanoparticle. That's a good question, actually. I think we'll have to be careful about looking at lines that don't have that flow GFP, but we have, here's the thing, we have flow 2 that we know, and flow 3 that we know goes into in this aggregated form here. Yeah, so anyway, I think that's a good point. I will have to keep that in mind.
Thank you. I think there is an online question. Shin, do you want to? Shin, do you want to ask your question?
Can you hear me now?
Yeah.
Hello?
Yes.
Okay. Yeah. My microphone was not set correctly. Sorry. So yeah. So it sounds like the flow proteins can form kind of like these liquid droplets or like aggregates on their own without like assistance of other like proteins, just based on the changing in the water potential. So, but then do they kind of interact with other proteins, either to kind of like, like separate them like from the cytoplasmic, or like kind of like protect them from during maybe the dehydration, so that they can be used later when the water comes in? Have you done any kind of like isolation or IP to see, if there's other protein kind of being absorbed into these liquid droplets?
Yeah, that's a good question. We have done pull downs
Okay.
of rehydrated seeds that, rehydrate mature seeds. And we don't really pull down anything other than HSP 70. I don't know if it's really meaningful or not. But it might be meaningful. I mean, they are known to disaggregate liquid droplets, aggregates. But what would be more interesting is what you actually mentioned. So during development, when the seeds are dehydrating, what's happening? What are the things that this might be interacting? We don't know that yet, but that's actually doing the pull down assays during seed development would be interesting, actually, as the seeds are drying out, or like if we are able to extend the rehydration period.
Right.
To try to do pull downs at this early stages. That might also be interesting.
So have you ever seen like how long does it take the seed to form these droplets after imbibition?
Yeah, so that's another thing we want to do using sort of fluidic device to actually observe, in real time the granulation. We haven't managed to do that yet. But it's one of the things I've been asking, I've been sort of advocating for a very long time. That's why I'm kind of laughing. But, they form granules like as soon as you dissect them and put them in water. So
Yeah.
I've been told like seconds, but
Okay.
whatever. What does that mean?
Yeah.
So anyway.
So do they?
They're fully soluble proteins. You don't have any membrane.
It's soluble, yeah.
Fully soluble.
Yeah.
So except for the one organism, does any other, like in any other species, does the phloem like kind of like relocate into the nucleus?
Oh, that's a really good question. So in Arabidopsis, it does, very
Okay.
transient. It's actually in the cell paper. The cell paper has about 5 times more data than I've just shown you today.
Yeah.
And it's one of those crazy 2-year publication process going back and forth. And every time you add more results, and then in the end, you know what they said? Bring down your 150-page supplements into 7 figures. Your supplemental figures cannot be more than your regular figures. It was just like, it was kind of, anyway. So it's really interesting that the protein goes into the nucleus transiently, at sort of in the middle part of the water potential curve.
Oh, okay.
And we don't know what that means, actually. And there's a very strong nuclear exclusion signal in the protein. And so it keeps it away. It's very important for some reason for the protein to be kept away from the nucleus. Okay, this nuclear exclusion signal is super strong. It's only 20 amino acids long, and it's like really strong. So I think it's got some role in the nucleus.
Yeah.
And I actually didn't share, I skipped over this thing, but I'm gonna actually maybe just 1 minute. We asked what are other proteins that behave like phloem, like that they have this prion-like disorder regions, and they're really most abundant in the seed. We found like these number, I think it's 15 proteins. And you can see they're mostly, other than flow, this unknown thing is phloem. It's essentially either transcription factors or RNA binding proteins. And you have things like ABI5 that's known to involve germination. So, that's another direction that we could go, is maybe phloem is acting as a novel type of transcriptional regulator for seed germination.
I just want to add, there is a recent paper come out last year about tau proteins, the one in Alzheimer. They actually found to aggregate and form bodies with RNA.
Yeah. Yeah.
So there is a cranium structure. It was a big discovery.
Yeah, I think all of
And-
these,
And
Yeah.
so do you think you have any nucleic acid, like a messenger RNA binding to them? Do you know any info?
we don't know what are, if, we haven't looked for RNAs in the granules.
Because that's what the key
Yeah.
came out in Science and Nature, I think, last year about tau proteins.
Yeah,
That they
yeah,
found
yeah.
associated with RNA, and that was the core of nucleation and forming
Yeah, yeah.
the filaments.
Yeah, yeah.
So I wonder, especially they have a history of overlap with transcription factor and RNA binding proteins. I wonder if you have something similar.
Yeah, we haven't looked for RNA, but we, it might very well be, that there could be interesting RNAs, that could be important. Yeah,
So they might be part of those bodies.
Could
That's
be.
what I'm trying to
Yeah,
say. So
yeah.
we found that the tau fibrils, the RNA is actually being part of those
Yeah.
fibrils. And
Right. Yeah, could be. That would be really interesting to look into. This is another direction that we'd like to go into.
Thank you.
How did you fish them out from transcription regulators? Because you initially showed that you have, like, you selected a class of 200 genes from transcriptional regulation, right? And then you narrow it down to the
Yeah,
flow.
Yeah.
How was your workflow? Like, how did you narrow it down to that particular?
that's a good question. So we actually screened all of the lines, like 60 or 70% of the 200 lines that we had knockouts for, and looked for phenotypes. And this was one that actually had germination phenotype.
Okay.
That's why we decided to work on
There are
it.
200 lines.
Well, we had 200 genes that were identified from our initial screen, and then we got the knockout lines of the salt lines. It wasn't 200, it was maybe 60%. So we then, after we got down that 200 genes, we just looked for phenotypes.
Okay.